BRC prides ourselves in being an emerging, collaborative, and multidisciplinary research presence. The focus of the BRC is the study of biomolecules with emphasis on proteins and protein interactions. The BRC represents a comprehensive collection of instrumentation and facilities ideal for the characterization of biomolecules and their role in a variety of biomedical and environmental processes. Partnerships exist between the Center in Idaho, Bryn/Embry, U of I, ISU, the Boise VA Medical Center, the College of Idaho, and Northwest Nazarene University. To support current and future collaborative science, the BRC provides seminars, training workshops, and other networking opportunities. The BRC is supported by funding from National Health Institutes, National Science Foundation, Murdoch Charitable Trust, Lori and Dwayne Stuckel, and the Idaho State Board of Education. Some grants include programs that support student researchers. An example of this is Embry, which is a paid research experience that provides hands-on experience in the field. Here's some more information.
You saw it?
Yeah, now I think it's the center is to facilitate multidisciplinary research and research training programs. Our goal is to increase the level of biomolecular research achieved at Boise State University, with opportunities for students and faculty members alike. The BRC offers instrumentation compatible with many types of research, ranging from molecular biology and tissue culture, histology, biomolecular characterization, imaging, and mass spectrometry. A few examples of instrumentation in the lab include CO2 incubators, which provide an ideal environment for cells to grow. The biosafety hood provides a sterile environment to keep cells free from microorganism contamination, which is critical in cell culture experiments. Confocal microscopy is a powerful instrument that creates sharp images of cells and tissues. Micro-CT scans and generates high-resolution images on 3D structure of a variety of samples, such as bone, teeth, cartilage, and lungs. And mass spectrometers are used to identify molecules based on their size and measure their quantity in samples. Biomedical research is a growing field with opportunities to improve human health. As a Bronco, you will have amazing resources to make your college experience truly life-changing. If you are curious about learning more on all things biomedical, please reach out to us. No matter where your journey takes you, we wish you success. From all of us here at the BRC, thank you for joining us today. Go Broncos.
Okay, I'm going to stop sharing.
There we go. Hi, Julie. Hello, Troy.
Hi,
Mindy's here as well.
Hi, Mindy.
Oh, hello.
Mindy, are you a admitted Bronco ready to start in the fall, or are you thinking about Boise State?
Yep, I am enrolled, so I'm pretty excited.
That's good. We're glad you're aboard.
Thank you.
So you got to see a part of a video that was about the Biomolecular Research Center. And now I'd like to share, if I can, I'm going to share a video from, I think it's 4 of our undergraduate researchers, and they just finished up this research project.
Okay, yeah. I would like to see that.
Let me check a couple of other things first before I start that. Okay.
Hi, everyone. This is Grace, my group partners, Darren, Izzy, Karen, under the supervision of Dr. Julie Oxford, and we're presenting the Baller-Gerold syndrome today. Baller-Gerold syndrome, also known as BGS, is a malformation of the cranium, as well as disfiguration or incomplete or complete absence of the distal zones and upper limbs, pretty much like your arms and thumbs and digits. The only treatment currently is surgery, which is risky, and that can also permanently damage mental functions. Also, there's effects from BGS on the cardiovascular, respiratory, and immune system. This creates a complex of burdensome syndromes. So this further research is important because we could try to improve the life of those affected by BGS and hopefully maybe find a cure one day. BGS is incredibly rare. Worldwide, it's less than 1 per 100,000 people are diagnosed, and there has been less than 40 cases disclosed in medical literature. Some of the clinical descriptions include the major abnormalities of craniosynostosis, which is the joints between the baby's skull fuse prematurely. This results in a misshapen skull. Other minor abnormalities include an anal or urogenital defects, central nervous system issues, and mental retardation, but those are more rare. As you can see in our first figure for A and B, it's showing the dislocation of the thumb, which is a limb abnormality. In C and D, it's showing that at the top ridges of the skull on the frontal view, the skull has prematurely fused, which ends up making it hardened and then hard for the brain to continue growing during development. The other image shows the back view of it. The etiology is that this is a genetic disease that's caused by a mutation in the RECQL4 gene. This gene encodes for the RECQL4 proteins, and shortage of this protein is known to prevent DNA replication and repair, so it has a lot of downstream effects.
So moving on with the RECQL4 gene mutation, if we look at figure 2A up in the top middle of the poster, we can see that the specific deletion is exons 13 through part of 18 on that RECQL4 gene. And it shows you actually down to which nucleotides are mutating or deleting from the genome. Then moving on to figure B, we have kind of a schematic showing how RECQL4 interacts with other proteins to help in double-stranded break repairs of the DNA. And on the right side of that figure, you can see that if you lack the RECQL4 protein, you're not able to repair your genome and you're going to have genome instability. And then with figure C, 2C, this is PCR showing the pattern of deletion in a patient. So on the far right, you can see that there's no exon 13 through 18, but the father, mother, and sister of the patient all have those exons to have a functioning RECQL4 gene.
On the next section, differential diagnosis, we use a Venn diagram to compare the similarities and the differences of BGS with other similar syndromes such as Rapidolino syndrome and Rothman-Thompson syndrome. And out of all the symptoms present that compare and contrast these syndromes, the two main factors that differentiate BGS from the other two are the presence of craniosynostosis. As Grace said, it's the premature joining and fusion of the skull bones in the baby, and the absence of cataracts. And under antenatal diagnosis, there are a few ways to diagnose the presence of BGS. The first one is using ultrasound to measure or to see the presence or absence of the skeletal parts of the skeletal system, such as the radial, the ulna, the phalanges, the carpals, metacarpals. And another method would be amniocentesis, which is when you extract the DNA of fetal cells that are present in the amniotic fluid and doing testing with it. And the third method would be doing a chorionic villus sampling, which is when one analyzes placental tissue that is extracted either transcervical or transabdominally.
Okay, so some of the management treatment, initially it's mostly consultations after the initial diagnosis. Typically you first do dermatology to evaluate for possible poikiloderma, which is shown in figure 4. That's a severe case of it. Also, a clinical geneticist or a genetic counselor to do comprehensive genomic testing and really see if the patient the symptoms they're having is consistent with the BGS. Orthopedic surgery for possible extremity functions to see if they could do surgeries to repair that. Neurosurgery or a craniofacial specialist to evaluate for craniosynthosis. Oncologists to evaluate for possible cancer development, since patients with BGS are at higher risk for it, and which is why they end up doing preventative measures to prevent things like osteosarcoma, which is a type of cancer, and lymphoma. Limitation of UV radiation just to prevent for possible skin cancer, especially if they have poikiloderma, that puts them at a higher risk for cancer. So treatment options, there is no cure for the actual syndrome. It's only treating the symptoms. So surgery, like it was initially said, to treat the craniosynthosis, if it's bilateral, typically this is usually performed before they're 6 months of age for the best operative results, and pellicization of the index finger just to help with functional grasps. Overall, based on the case discussion on this information, the general age of patients that undergo treatment are between the ages of 2 and 4. And an unresolved question in this disorder is generally leads to the death at an early age, or if treated early enough, patients can live to adulthood. Future research can be performed to define additional signs and symptoms that distinguish the BGS from similar syndromes, in addition to the broad phenotype of the craniosynthosis, and which involve the RECQL4 gene. Further studies on effective genetic treatments such as CRISP may be beneficial. That's it.
And now we have a few questions to answer from our classmates. The first one is from Molly. She was asking in regards to our figure 2C, if patients have no expression of any exons, is it possible to have some expression but not all of them? What would be the result of having a partial expression? Also, is there any treatments before birth that can be looked into, or the possibility of early diagnosis and treatment in the womb?
So to respond to Molly's question, for the first part is yes, mutations and deletions in exons or the entire gene itself can vary from patient to patient. And the different combinations produced by the different deletions can cause a wide spectrum of symptoms denoted to BGS, such as some of the major abnormalities or the minor abnormalities. There's been case studies of patients that have parts of the exon 15 deleted or the entire gene itself, and they present different symptoms that vary from T-cell lymphomas to compromised immune systems, cardiovascular, respiratory, or even skeletal abnormalities. So depending on how much is deleted and what is deleted, the symptoms will vary. And since the limited research on BGS is very limited, this restricts the research of effective in vitro treatment or medication. But however, the earlier the detection of BGS, the earlier parents can prepare to intervene as soon as the child is born. And the sooner that the surgery is done to repair craniosynthosis, the better the outcomes, as Karen mentioned earlier.
So our second question is from Chrislyn. She asked for our poster, if it can be treated, if the syndrome can be treated enough so that patients do live to adulthood, what would their life look like? Would they need constant care? Would they need to have ongoing medical procedures? And possibly is there a chance, higher risk of cancer or any way to help with the bone abnormalities?
Yeah, so life for individuals affected by BGS is going to be a little different for every patient because there's a lot of varying levels of severity that this disease will present with. So, for example, if a patient had extreme underdeveloped upper limbs and cranial deformities, that patient will need a lot more attention than a patient who just has, some thumb abnormalities or maybe the pachyderma. So yeah, it kind of is taken on a case-by-case basis because there's so little known about this disease. And there's also not a lot of actual cases known about or studied very well. So it's really taken on a case-by-case basis, and the healthcare professionals are treating for symptoms of the disease.
I thought I might pause the video to see if anybody else that's in the chat with us had any questions about this research or how these students got involved in research.
Who do we have on the call? I see, let's see. I see Mindy.
Don't call me, please. I'm not prepared with it.
I see Michaela's here. Welcome back.
Hi.
Michaela, what are you interested in?
Like professionally, like goals wise?
Yeah.
Well, I'm interested in the future becoming a forensic pathologist. So that sort of thing, like autopsies and pathology, but pretty much anything biology wise.
That's great. How about you, Mindy?
Well, I want to go into the health field. I think I want to become a family physician.
Good, great. Let's see, who else is on the call?
I'm trying to get Jazzy unmuted right now. Let's see if we can have any luck there.
Can you hear me?
Yes.
Okay, I'm unmuted.
Great. Hi, Jazzy.
Hello.
So what are you interested in?
I'm interested in biomedical sciences, more specifically like microbiology or molecular biology.
Yeah, great. Well, there are lots of opportunities at Boise State in the biology department. Several different faculty members do research in microbiology. Troy Rohn is on the call. Troy is a faculty member in the Department of Biological Sciences doing Alzheimer's research. If you have any questions for him.
I'll just chip in here and say I'm an advisor for biology students, and I have talked to all 4 of the students doing that research on the Ballard-Gerald syndrome. And all of them have mentioned it. All of them have talked about how exciting it was to, to be digging into a research project where they felt like they were really making a difference. So it became like an exciting part of their undergraduate experience.
And some students do research during the academic year while they're taking their classes. So they balance their research within their courses. Sometimes they're earning credit for the research. Sometimes the research comes in the form of a paid internship during the academic year, but also sometimes during the summer. So we have some biomedical research internship programs available that run for 10 weeks during the summer, 40 hours a week, and students are paid a stipend. They get to present their research at a meeting at the end of the summer and interact with other students that have been doing research all summer long. I was trying to remember how many students across campus do research every summer, and I think it's in the hundreds. There's a pretty exciting summer research community every summer. So you get to be with a lot of other students that are doing the same kind of thing that you are doing.
Yeah, and I'll just chime in. This is Troy. This is the first time I've seen Julie's poster here. Like, two of these students I had in my classes. Izzy's one of the students actually is I know is on the track team, and so he's actually a Division I cross country runner. And most of the time when we get the athletes in our biology curriculum, they're really good students because they actually have to learn how to manage their time, and they're very, very motivated. So it was good to see him. And I also, I know Darren very well too. I think actually he's applying to medical school, if I'm not mistaken, right now. And I usually get a lot of those students because I teach neurobiology and pharmacology, which are two courses that most of the pre-medical students take, not just pre-med, but physician assistant. There's veterinary students. We get pre-dental students. So a lot of the students don't actually get involved in research, but a lot of those that are interested in going on to, say, medical school need that for their resume. So we get asked a lot. And it can be competitive, to be honest. It's not like you just get to choose what, like you can go into a lab. Like normally most of the students have to be pretty persistent. So that would be one thing I would definitely advise if you're interested in doing research, is that you need to be persistent in finding a good spot that's a good fit for you.
Those are all really good comments, I think. And I think, so here you see a little bit of developmental biology research, and Troy can tell you about neurobiology research, but also in the department, we have laboratories that are working on cancer biology, vaccine development, liver health and liver physiology, Parkinson's disease,
RNA evolution. And then what's Alan work on?
Oh, notch signaling and the cardiovascular system.
Yeah.
We have one investigator that works on the blood-brain barrier. Really fascinating work.
Most of the students, though, it's important. A lot of them, we don't usually see them in the labs until they're juniors or seniors, because it's important that they get a sufficient level of background, scientific background, in some of our core courses. And a lot of us like to get to know the students first before we accept them into the lab. So usually, at least my students are usually juniors or seniors before they have an opportunity. Now, that's not always the case, but I tend to like the students are a little bit more mature, and they're going to be more dependable, and they've had some background, so they know, for example, what PCR is, and what when we say protein or gene, they have a very good understanding of all those basic biological principles.
But if you, in your first and second year courses, if you find a topic, if you guys cover a topic that is really of really strong interest to you, I would encourage you to reach out to your professor or your lab instructor and let them know that you're particularly interested, and if there are things you should do next, or people you should talk to, or some additional work that you can do in that area, they can help guide you to some interesting possibilities. And then Clay, our academic advisor, Clay meets with all of the students, and Clay can also alert you to opportunities like the VIP program for research. There's a vertically integrated projects program at Boise State that gives first and second year and even third year students a way to get their foot in the door to research. And you earn credits, you can repeat the class throughout your college career, and then every year you continue in that type of program. Your effort and your responsibility and your participation increases and increases until your junior and senior year. You're one of the more experienced individuals, and you end up teaching the first year students in a way. But it's a very team-based approach to doing research.
Julie, was that
so keep your eyes open for
that? Jenna Klingerman?
Yes, she was in that. Yeah,
she was in your VIP lab for a long time, I think.
Yeah, she was, and now she's at Purdue, I think, and in grad school.
Yep.
Yeah.
So, and I think there are other opportunities if you don't want to go into the lab, like a lot of times we write review articles in our field. And for example, Julie's daughter just helped me. She was an undergraduate, helped me write a review paper that just was published. And that's a really great experience because you learn about scientific writing and how to write and how the whole publication process. And of course, for her to get a peer-reviewed paper, that's like the holy grail of that just made her so much more competitive. Is she applying right now for medical school, Julie?
Yes, yeah, she's taken the MCAT and she's applying, doing some volunteering right now. So yeah, publication is really, really great. Yeah.
So there are other opportunities, but it depends on it. It's not always has to be that, you know, you get into a wet lab, and there are modeling and types of people that just do computer type analysis, data analysis, or just writing and a review article on, for example, a specific topic in Alzheimer's disease. That's what Alex did with me. So those are also really important opportunities that students can gain in terms of research experience.
Bless you.
Do you guys have any questions for us?
So I have a question. So you mentioned a summer internship program for research. So how do you get involved in something like that?
So that's a really good question. So every year in late fall, we advertise and try to get the word out to everyone across campus about this opportunity. And then there's an application process. There are a few essays that are included in that application. You know, we want to know what your goals are, and if you've had any obstacles that you've overcome, if you've had any research experience before. And then we ask you to provide 2 letters of reference, and then those are all reviewed. And then there's a matching process between the mentor, the faculty member, and the student. And then the awards are usually made by March, and then you're ready to start in late May for the whole summer. So it's every year. Well, we do our best to get the word out, but if you're at Boise State and you haven't heard about these summer opportunities, just reach out, let me know, and I'll put you in the right direction.
Okay, thank you.
Sure.
And Michaela and everybody, I also have just included in the chat the website for the STEM initiative on campus. It's an office that helps coordinate a lot of these kinds of activities, research opportunities, including these summer research opportunities. So it's a great it's a great website and a great office to be in touch with. They will coordinate some of these opportunities for you.
And then I have a question. How does this all work with people who are going in as transfer students? Does that mean everything's sped up and like fast? Or I don't know, is it harder to do all the research and all these opportunities if you're a transfer student?
Maybe it is a little bit harder, but we definitely encourage you to get right on it because you have had a lot of the general science, a lot of times, the general biology, general chemistry, physics, maybe. These things are at least in place and you have that confidence. And it's like I tell students daily, confidence is part of it, is knowing that you're capable of this. So you'll have to get out and start doing that networking that Troy was talking about, and get on every opportunity you can and start asking questions, going to office hours with faculty, getting involved. Ask me questions. I have a huge document that it's not that pretty to share on the screen because it's about 3 pages of links, but it's kind of like just different ways that students have got involved. And sometimes you end up involved in a research project that isn't specifically what you want to do for the rest of your life, but it definitely is teaching you skills that are really, really close and can be useful for the next step. And that's why we're trying to get everybody out of the classroom and into research opportunities, because it makes you, not only you'll be more satisfied with your undergraduate experience, but you'll leave here with the skills and the kind of background needed to move on to the next higher level.
You know, and another thing to keep in mind when you're a student here at Boise State is that every week there are multiple opportunities to attend seminars, and the seminars are open to anyone, but sometimes it can be a little intimidating because they're delivered at a pretty high level. There are experts coming in that visit our campus to talk about their research. But I think it's important to remember that if you just go in with an open mind and just absorb all the terminology and information, you may not understand it the first seminar you go to, but every seminar you go to after that, you'll get a little bit more and a little bit more, and you really will become fluent in scientific terminology, and you'll get a really good idea of why people ask the scientific questions they ask, and how the techniques work, and how they put it all together. And so I would encourage everybody to go to seminar. So I don't know.
And then I had a question, but I just lost it. Oh, you said to get in to learn about all the research opportunities and like internships to talk to any faculty member, or is there like a website we could go to, like the STEM website, go there for all the opportunities?
Yeah, I'm also gonna, just now I thought I'd pop up the website for the VIP projects that Julie mentioned earlier. I think the biology department has 6 VIP projects running right now. There's probably a total of 30 of them on campus, and they're pretty exciting. Like, students are getting out and doing some really unique projects. Let me sort out that website for you.
Well, and I would say that the best, if you're interested in doing biomedical, the best thing to do is just get on the biology website and look up the faculty and see what they're doing. I mean, most of us have a page, and we all give descriptions of our research, and you're not going to you can see the publications, and they're you may not get a lot of the publications, but I mean, that's how a lot of students learn about. One way they learn about the kinds of research that we do, but also in the classroom, once you start taking classes, most faculty will talk about their research one way or another during the course of the semester. And so that's another way that you can kind of learn about opportunities.
Thank you.
Did everyone get that those two websites I sent to you? Good. That's just the start. There's so many things, so many ways to get involved, and talking to your advisor is one of them, because I'll help you sort out anything you've missed on that list. I'll remind you to keep going until you find something exciting to get involved with.
What about biology club or any of the other clubs, Clay? Do you think those are good links for?
Absolutely. Grace and Darren are both officers in the biology club, and it's certainly been a way, not, you're developing leadership skills, you're developing interpersonal skills, those kinds of things, but also just really a way to involve yourself with other students, and you get like this different perspective, and it's also a great networking tool. So the biology club invites faculty to come to like special presentations and talk about getting involved in research and how to do it. And they have sort of professional development events where they kind of help you craft the best letter of intent or fill out an application, things, sort of practical skills that you learn. And the biology club also does a lot of fun stuff. So that's important. We are just about out of time. I want to just give you all a chance if you have any other questions. One thing you can do if you would like me to follow up is just send me a chat with your email address and any question, and I will get back to you that way. You're certainly welcome to just look us up from the biology department website as well. We're all checking our email very frequently these days because that's all we have to do, basically.
And you guys are welcome to reach out to me too. I'm happy to answer questions. And if I don't know the answer, I'll hunt it down for you. So.